This is Joseph Kazari, instructor at Mississippi College, and this is the mini lecture for Lab 4, the cell, microscopy, osmosis, and diffusion. First thing, a little couple points is we're not going to talk today, right now, about microscopy. You need to be familiar with the parts of the microscope, which can be found in your manual. I believe it is, I think it's about page 41 to 43. That's something you need to be aware of. But with osmosis and diffusion, we are discussing how items get in and out of a cell. It's kind of a big deal. All right, we need our body needs to take in things and get rid of other things, items. Right, the first type of movement is referred to as diffusion. Diffusion is the movement of molecules from a high to a low concentration. Right. When we discuss molecules, we're talking about solutes. Solutes can be ions, solutes can be gases, solutes can be little molecules of sugars. They can be amino acids or so on. Usually ions are not molecules, but nonetheless, when we talk about it's the movement of solutes would probably be a better word, but they both work for what we're dealing with today. And the big thing is we're going from a high to a low concentration. What that would look like is, we're doing drawings here. Of course, you can find these little animations online. But let's just imagine you have a little beaker of water. And in this beaker of water, you have a, let's say, a green sugar cube. This green sugar cube is at a very high concentration. As the water gets into it, the sugar, or the dye, whatever you want to think of, is starting to bump into each other. It's starting to work its way further out, further out, further out. It's going from this high concentration here to a low concentration elsewhere, all right, until it gets into an equilibrium where it's all green water. All right. So from a high concentration to a low concentration. Now about diffusion. First of all, this little bumping, right, this little natural bumping, Brownian motion is what it's usually called. It does not require energy. This is how gases get around naturally. Molecules in the air get around naturally. If you spray perfume in the corner of the room, someone in the other corner of the room or a little further away will smell it eventually when it diffuses to them. No energy is needed. Another nice thing about diffusion is we're going to have to test this using dialysis tubing. Now, the dialysis tubing is selectively permeable. Sometimes semi-permeable may be used. It means only certain things will get through this membrane of this dialysis tubing. In order to get it to work, you have to make sure it's wet, and then you're going to have to rub your fingers around it until it opens up. And then you can put whatever you want to in it as long as you tie or clamp off what it is. Well, this dialysis tubing, since it's selectively permeable, right, to small items. Right, very small, right, small substances. I'm not going to say molecules. For example, right, glucose. At this point, you should understand that glucose is just a small single, typically just a cyclic structure compared to starch. Put over here. Remember, that's a long chain of molecules, of these glucose molecules. And it goes on and on and on and on and on. Much larger, even though I drew it smaller, because each circle represents a glucose molecule. It could be ions. All right, salts, that is. Another small molecule you should be familiar with, H2O, water. Water should be able to get through that. So imagine when you put a fluid inside these dialysis bags, these tubing, and you tie one side off, water will be able to leak out. So you have to be careful with that. Let's see what else. So starch, if you put it in a dialysis tube, it probably would not work very well. What you're going to be doing is you're going to take some dialysis tubing. All right, it's going to be like a small, flat little sheet of paper, a flat little tube, excuse me. And you're going to put a clamp. You get some little blue clamps in front of you, or you will have that. You're going to put whatever substance you need to in there, then you're going to clamp the other side off. And you're going to have your glucose solution, starch solution, whatever it is on the inside. So that's diffusion. Next thing, after discussing diffusion, we need to discuss osmosis. Osmosis is the diffusion of water. Right. So if you know what diffusion is, right, it's the same thing. Diffusion by itself is just the movement of these solutes, ions, gases, sugars, little molecules, and other things. However, when we're talking about the water moving, we're referring to osmosis. Diffusion does not require energy. Therefore, osmosis will require no energy. Also, the diffusion is molecules moving from high to low concentrations, where osmosis, you're moving from a high to low concentration of water. For example, here's a beaker with a fluid, and we have some dialysis tubing, some type of semi-permeable membrane here. We have water molecules. Let's do that with, let's see, will water be X? And we'll let the, let's see, water the blue X's. And we will let these little orange or red circles be our solutes. Now for this example, these red solutes are too big to cross that semi-permeable membrane over here, right, even though they want to due to diffusion. But water can go through that semi-permeable membrane. Right. We have more water on this side than this side. Most of the time with dealing with osmosis, you talk about water wants to go to where there's less water. You can also say this: water wants to travel to where there's more solutes. All right. So here's a question. Which way will the water, in our example here, flow? When water is moving, we typically refer to this as osmotic flow. And the answer would be the water would go in that direction. And sometimes the water level on one side may increase while the other one decreases on the other. So it's going to where there's a higher concentration of solutes. So still talking about osmosis. All right. This is a solution of solutes. Right. Does it have a lot, some, or very few solutes? Well, solutions can affect our cells in 3 different ways. The first way, or the first type of solution, is referred to as a hypotonic solution. This is in no order. The second will be referred to as a hypertonic solution. And the third is going to be an isotonic solution. Just based on the name, right, the fluid that is surrounding our cells has less, hypo means less, has more of something or is equal of something. When we see this, we're referring to these solutes that we see here. Right. Let's start with let's go hypotonic, excuse me. Right. We see in this example, we have a cell, and you know what, let's go with a red blood cell. Red blood cells are good examples because once you eat something or drink something, it's going to be in your blood cells, it's going to be, in your blood within anywhere from 30 minutes to a couple hours. Let me try to add some color to this. So you got a solution, and in this, of course, there's water. Right, this is a hypotonic solution. So we got little solutes. We got little salts, sugars inside the cell. Outside the cell is the solution. And what we can see here is I'm just putting an X to represent some outside solutes. Very diluted. So where would the water go? Well, the water is going to, let's get this set up. The water is going to move into the cell because water wants to go to where there's more solutes. So that's going to dilute these a little bit, causing this cell to either swell or even burst. If this happens inside a red blood cell, this is referred to as hemolysis in red blood cells. This is the equivalent of being overhydrated. This condition can occur usually if you just drink a lot of water. Maybe you're an athlete and you're drinking nothing but water. You're sweating. You're losing electrolytes, but you're only replenishing your body with just plain water. what happens? It causes those cells to swell. All right, you shouldn't just replenish water, but you should replenish the electrolytes that are out there. Right. Hypertonic solution. In a hypertonic solution, we have solution. Let's stick with our red blood cells. In this case, we're going to have our concentration inside the cell, but we have too many solutes outside, hence the hypertonic portion. Right. By looking at this, right, where's the concentration of water higher? Well, yes, we know there's more water, but concentration, it's a comparison to a medium. Right. There is more water. It's more diluted inside the cell. Well, water wants to go to where there is more solutes. So it's going to leave the cell, causing your cells to shrivel up because the water escaped trying to dilute this outside solution. This is called shrivels up. I'm supposed to say shrivels, but it's plasmolysis. All right, this cell is plasmolized. Inside of our red blood cells, it's called hemolysis. Nope, sorry. Look, it's right up there. It's called crenation in red blood cells. Right. In the last situation, oh, by the way, this situation can occur because maybe you're eating too many salty foods. Right. When you eat too many salty foods, your cells being shriveled up, this is the equivalent of you being dehydrated. When that happens a lot inside your body, usually that causes thirst. Now, the isotonic solution, iso means equal. I think a lot of people should learn that in trigonometry. But in this case, the cell has a certain concentration inside and a certain concentration of solutes outside. All right, they're pretty equal. It's in equilibrium. Water can leave on its own, but it can also enter on its own because these cell membranes are usually permeable to water, if you haven't caught that yet. So what will happen? Nothing. It stays the same. It is in equilibrium. And this is something that you're going to work with today. You are going to work with, I believe it is some onion skin. So in plants, when you're looking at the microscope, you're going to see a lot of little squares. These little squares are the cell walls. Inside the cell wall, that onion, it's more of a red onion. All right, so I'll be using the red color here. Inside of this is inside the cell, there's plenty of pigments, which gives it that red color. So the first thing is, let's stick to this one, isotonic. What happens if we put this cell in an isotonic solution? Isotonic would be just plain tap water. That is straight from the sink. It has some ions in it. It has little things that some are never getting pured out that there's some chlorine in. So when it's all said and done, it should look the same. Not much difference. Oh, by the way, when you're drawing this, you have a circle. The best method of retention, a really good method of retention is drawing. When you have a circle on your lab manual and you are looking at something, if you draw something like a little square like this and you have to draw hundreds of them, obviously you can increase the magnification. Do not do that. That is ridiculous. Right? You don't have to draw all of them, but you can increase the objective. Right? Do not grab the objective to increase it. There's a little nose piece. If you grab those objectives, you can untwist them. You can pull them out. But you don't have to draw every single one. And the highest magnification, you should be able to see at least like 10 or so. And I'm just drawing 2 of them, 3 in this case. After that, what we want you to do, and I'm going to do something a little different. So we're in isotonic solution. We are going to use that same cells, and we are going to add, we're going to put them in a hypertonic solution. Hypertonic solution is salt water. You should have some at your station. And the equivalent of that is what we see over here with this little red blood cell. And you should notice that this is the cell wall, keeping it stiff. The cell membrane is more flexible. The cell membrane, which is red, is going to shrivel up. Right? Because the water left that cell membrane. Right? You draw it. Hopefully you're observing this. We're not going to make 3 different sets. We can use the same onion skin for one than the other. Now, what we're going to do is you are going to put it in a hypotonic solution. Now, if you come over here, a hypotonic solution is water that doesn't have much else in it. Clean water. So we're going to be using DI water. All right. It's referring to deionized. It might say distilled water. It just means it's very pure. It's very clean. What we're going to notice there, and I'm just going to draw from another angle. Right. According to this, water should move into the cell, and that should be shriveled up a little more. Excuse me, should swell up more, and it may burst. But here, the water is going to move back into the cell. So what are we going to see? It should go back to normal, if not more tight against the cell wall. These are things you should observe. Now, that's not the only type of movement in and out of a cell. That's the only type that we can discuss here in the lab, or that we can observe in the lab, I should say. There are others. For example, 2 other methods of movement would be facilitated diffusion and another one would be active transports. Now, for facilitated diffusion, in the name, as it implies, it's diffusion. Diffusion goes from a high to low, and it requires no energy. So what is facilitated across a membrane? Typically water, right? Some small molecules, maybe. They usually diffuse through channels. how that would look is here's a membrane. Right, but this membrane has, they don't have holes in them, they have proteins. Protein, there's a protein. And here's some, let's say we got maybe some little red water, small items. Right. Let's put a sugar molecule up here. Right. And then maybe we might have a very large, let's put a protein there. Now these channels, or pores, they could also be called. Right, small things like water can go through. All right, this protein is only made for, let's say, water. It's not the right size for this glucose. It's definitely too small for that protein. Protein cannot go through that. As for something like glucose, well, this protein, this is like it looks like two, but it's really supposed to be like a tunnel. All right. Where this protein molecule will allow small items, specific items, because remember specificity, it only allows certain things or reacts with certain things. This only allows glucose to come through. Right? But that says if there's more water on the outside, it will come in. If there's more sugar on the outside of the cell, it'll come in. If there's more water on the inside, then it'll go out. We just described that up here. Active transport, right, there's no word of diffusion in here. This one uses energy. And what it does, it doesn't go, it's the opposite. No energy for diffusion, uses energy down here. Diffusion goes from a high to a low concentration. This is the opposite, going from a low to a high concentration. A very good example of this are the ion pumps in your cells. Here's our new cell. Here is a green ion pump, also known as the sodium-potassium pump, because those are ions, right? Right. Well, in this case, we have high concentrations. We have high concentrations of sodium outside the cell, and we have high concentrations of potassium inside the cell. These brackets represent concentration. But what happens is that this ion pump will find potassium and it will bring it inside, going from a low concentration to a high concentration. It's stuffing the potassium in there. Same thing with sodium. It'll also collect sodium, right, removing it, taking it to the outside. There's hardly any sodium on the inside, but it's going to take it out from a low concentration, go into a high concentration. And this requires energy, usually in the form of ATP. That's your cell's currency. But those are items we're not going to observe here. Now, once again, make sure you know your parts of the microscope. And a good idea is for this lab, is to start with procedure 6. Get that one started, and then go back to procedure 1 and while you're waiting on procedure 6. All right, this is Joseph Kazari signing out.